Please explain in detail the molecular genetics, such as nucleic acids, transcriptional regulation, prokaryotes, mutations, and viruses. Molecular genetics is the study of genes and their functions at the molecular level. This field encompasses various processes that involve the storage, replication, and expression of genetic information, as well as the mechanisms of mutation and viral genetics. Here, we will provide an overview of nucleic acids, transcriptional regulation in prokaryotes, mutations, and viruses. 1. Nucleic acids. Nucleic acids, DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are the fundamental molecules responsible for storing and transmitting genetic information. DNA serves as the genetic blueprint, with its double helix structure composed of 4 nucleotide bases: adenine, A, thymine, T, cytosine, C, and guanine, G. These bases pair with each other through hydrogen bonds, AT and CG, to form the double-stranded DNA molecule. RNA, usually single-stranded, is involved in the translation of genetic information from DNA to protein synthesis. 2. Transcriptional regulation in prokaryotes. Transcriptional regulation refers to the control of gene expression at the transcription stage. In prokaryotes, this regulation is crucial for adapting to environmental changes and conserving energy. 2 primary regulatory elements are involved: promoters and operators. A. Promoters. These are specific DNA sequences recognized by RNA polymerase, the enzyme responsible for transcription. Promoters are crucial for initiating transcription, and their strength can influence the rate of transcription. B. Operators. DNA sequences within or near the promoter region. Operators bind regulatory proteins called repressors or activators. Repressors block transcription by binding to the operator, while activators enhance transcription by interacting with RNA polymerase or other transcription factors. One classic example of prokaryotic transcriptional regulation is the lac operon in E. coli. The lac operon contains 3 genes involved in lactose metabolism, which are expressed only in the presence of lactose. In the absence of lactose, a repressor protein binds to the operator, preventing transcription. When lactose is present, it binds to the repressor, causing a conformational change and releasing the repressor from the operator, allowing transcription to proceed. 3. Mutations. Mutations are alterations in the DNA sequence that can result from various factors, including errors during DNA replication, exposure to mutagens, or spontaneous changes in the DNA structure. There are several types of mutations. A. Point mutations. A single nucleotide base pair is replaced, inserted, or deleted. Point mutations can be silent, no change in the amino acid sequence, missense, change in the amino acid sequence, or nonsense, premature stop codon. B. Frameshift mutations. Insertions or deletions of nucleotide bases not divisible by 3, which alter the reading frame of the codons, often leading to non-functional proteins. C. Chromosomal mutations. Changes in the number or structure of chromosomes, including deletions, duplications, inversions, and translocations. 4. Viruses. Viruses are small infectious agents composed of genetic material, DNA or RNA, enclosed within a protein coat called the capsid. Some viruses also have an additional outer envelope derived from the host cell membrane. Viruses lack cellular machinery and can only replicate within a host cell. They hijack the host's cellular machinery to replicate their genetic material and synthesize new viral particles. A. Viral replication. Viral replication can be either lytic or lysogenic. In the lytic cycle, the virus infects the host cell, replicates its genetic material, assembles new viral particles, and ultimately causes the host cell to burst, releasing new viruses. In the lysogenic cycle, the viral DNA integrates into the host's chromosome and remains dormant, allowing the host cell to continue normal functioning. The viral DNA, called a prophage, can be replicated along with the host's DNA during cell division. Under certain conditions, the prophage can be induced to exit the lysogenic cycle and enter the lytic cycle, leading to the production and release of new viral particles. B. Viral genetic material. Viruses can contain either DNA or RNA as their genetic material. DNA viruses usually have double-stranded DNA, while RNA viruses can have single-stranded, positive sense or negative sense, or double-stranded RNA. Retroviruses, a type of RNA virus, use the enzyme reverse transcriptase to convert their RNA genome into DNA, which is then integrated into the host's genome. C. Viral evolution. Due to their high mutation rates and short generation times, viruses can evolve rapidly. This rapid evolution allows them to adapt to new hosts, evade host immune systems, and develop resistance to antiviral drugs. In conclusion, molecular genetics is a fascinating field that encompasses the study of nucleic acids, transcriptional regulation, mutations, and viruses. Understanding these molecular processes and mechanisms can provide valuable insights into gene function, genetic diseases, and the development of novel therapeutic strategies.